In this exercise, we're talking about looking at different slides under the microscope. Instead of making the slides yourself, sometimes you will use slides that have been commercially prepared. Why would you use these versus making your own? Sometimes the samples may be hard to obtain. Maybe you can easily get the sample, but it's hard to prepare the sample for staining for you then to view it. Most things you have to add, certainly in microbiology, you have to add a stain in order for you to be able to see it. Without a stain added, they're going to look like little tiny, almost like ghost-like specimens in there, and you're really not going to be able to see much, especially for bacteria, because you're going to see cells, whether you're talking about bacteria, whether you're talking about fungi, they're very small, and so it's going to make it a lot easier if you add the stain, but depending on what you're looking at, that can be hard to do. So oftentimes it's worth the money to go ahead and purchase prepared slides. So we have quite an extensive library of prepared specimens. We have things from certainly animal and plant specimens to look out, teaching anatomy and physiology. We have a lot of tissue samples, different types of cells, from skin cells to muscle cells, etc. But we have a lot of, in addition to that, of whitefish eggs, cells going through mitosis. In microbiology, we tend obviously to look at even smaller things. So we have fungal cells, we have cells of protists, which are typically unicellular eukaryotic cells, and then down to bacteria. Now, do we have slides of viruses? No, because viruses are so small, you cannot see them with a compound microscope. You would have to have an electron microscope, and we don't have those. Those are very expensive. It takes a lot of training to learn how to use them. So we just have the compound scope. This particular picture on the slide, this is bacteria. The TM means total magnification, so that's telling you the magnification that you're viewing this at. In this particular one, the cells have been, or the sample's been magnified a total of 400 times. And this is just to give you kind of a reference point. Each one of the little teeny tiny dots is an individual bacteria. So right here, these are just clumps of them. They're little tiny ones. So usually to view bacteria, we are going to go up to 1,000 times total magnification. But this is just showing you that's an example of one of our prepared slides. It depends on what you're looking at, to wanting to learn as to what magnification you're going to look at. This is an example. Once again, this is a total magnification of 400 times, so the same as the previous slide. This is yeast. Yeast is single cell fungi. So each one of these little circles here is an individual yeast cell. A little bit bigger than the bacteria, but still small. And then here's another example, one of our prepared slides. We do have a protist. One example of this is dinoflagellates. Once again, this is magnified a total of 400 times. But these, you can tell, are much larger than what the two previous slides were of those specimens. So in this exercise, when you do it, you're just going to be looking at a variety of different prepared slides. Notice when you're looking at it, the arrangement of the cells. Notice the size of the cells, because as I said, it's a huge variety. You can try to calculate what the individual cell size, remember from previous experiments. You know, to do that, you have to know your field diameter and then multiply by the number of cells that would fit across your field of view. So you can do that. As I previously said, oftentimes a stain is added for you to be able to more easily see your specimen, but notice the color on that.